So, thank you. So I will then talk about mucus, which is my main interest, and it's actually in the middle as being the host microbial interface, in the middle of what we're talking about in this conference. But first, I want just to introduce mucins because they are the main component of mucus. The main feature of mucins is that they have this PTS domain. A PTS domain is a proline, threonine, and serine-rich domain that is the anchoring sites for all the O glycans attached to these molecules that give them the properties of the mucus. There are two families of mucins. One is these gel-forming mucins that are transmembrane mucins that anchor to the apical surface of the epithelium and make up the brush border or the glycocalyx. And then there is this other type that is the gel-forming mucins that are oligomerizing molecules that are secreted and make up the mucus gel that is covering the epithelial surfaces. And if you look in evolution, the transmembrane mucins appear a bit later, while the gel-forming mucins is actually a very ancient system that we have preserved and that apparently is valuable for us in protection, I guess. So if we look at the gastrointestinal mucus, this is done in rats, and we can see that in the different organs here, we have different demands. The tissue needs different things from the mucus, and therefore the mucus layers have been solved in different ways or organized in different ways. In the stomach, where we ingest a lot of hazardous material maybe, we have the problem with the acid, and we have mechanical forces that are quite dramatic. We have an inner layer that is attached to the epithelium that is quite dense. This layer and this mucus is then made up by a molecule that is called Mac5AC, a gel-forming mucin. If we go further along the intestine or the gastrointestinal tract, we come to the small intestine. The demands in the small intestine are completely different. Here we need to be able to absorb nutrients during digestion, and we need the luminal material to be in somewhat of contact with the epithelium. We also here have a mucus layer, but this mucus layer is different. It might have a patchy appearance, or it could also possibly be, which has been shown by some people in EM pictures, a mucus blanket. But what I know is that this material is much, much more permeable than any material, for instance, in colon. So larger molecules are able to penetrate this mucus layer and get in contact with the epithelium. The mucus is primarily secreted at this site, just on top of the crypts, which of course is the area you want to protect. You want to protect the stem cells down in the crypts. And this also gives a different need for other systems as antimicrobial peptides and so on to help with the defense for the epithelium. Then we come to the colon, and in the colon, again, we have a different setting where we don't need such large molecules to be able to penetrate the mucus. It's instead small molecules like short-chain fatty acids and water, of course, that need to be transported in ions through the mucus layer. But the problems are that we have all these microbes, this enormous amount of complex microbes that we heard about a lot today, that we need to protect ourselves from. And we have solved this then, again, by having an inner mucus layer that is dense, compact, and attached to the epithelium. And this layer is actually not as the cartoon shows here, but rather thicker in the distal part, where we also have the most mechanical forces pressing the luminal material onto the epithelium. Of course, that makes us need a better protective mechanism of the mucus here. So that is my view of the mucus in the gastrointestinal tract. And in the intestine, the mucus is made up of the Mac2 molecule. So what do we know about the Mac2 molecule? Mac2 is an enormously large molecule. In humans, written here 5,200 amino acids, and it has got variable number of tandem repeats, so it can be longer or shorter in different individuals. It has these PTS domains in the middle that are highly O-glycosylated. And then in the flanking terminal regions, it's got cysteine-rich domains that have domain structure resembling the von Willebrand factor. And during biosynthesis, this molecule forms dimers in the ER, already then quite large. Then it is O-glycosylated, and it adds an enormous mass to the molecule. Finally, it is trimerized, making an oligomeric structure in the N-terminal part here, and at the same time, it's stored, condensed, compacted, and stored in these mucin granules. This is facilitated by low pH and high calcium. And then during secretion, so when this material is released, it is expanded enormously in volume. So about 1,000-fold at least, these compact molecules are expanded, making sheets that then can be structures into this mucus layer with these dimers and trimers. And if we look at the real material using EM pictures, we can see packed material in the vesicles, and during secretion we can see that we have more released material, it's more expanded, you can see the material there. And then outside of the cell, so we have the epithelium down here on this picture, you can see this stratified organization of the mucus layer when it's secreted. And you can also see that there is a density gradient from the epithelium out towards the lumen. So this stratified mucus is then formed with this inner layer that is really compact and dense, and it's adherent to the epithelium. This material also has an insoluble core, and it is protease resistant. I'm not saying that it's resistant to all proteases, but if you add trypsin, for instance, you do not completely dissolve the mucus. So it is resistant to a lot of proteases. While then it is converted, after a while or after this distance, it's converted to a loose material that is expanded in volume about 4 times with proteolysis, or at least that is part of the mechanism. And this material is then soluble instead. And if we look more into the composition of mucus, I found a lot of different molecules that could contribute to the mucus layers, or are found in these mucus layers. This approach did not take or detect small molecules like the defensins or antimicrobial peptides and so on, but it identified a lot of other molecules. One of the molecules that was identified was this molecule, Fc gamma binding protein, which is a quite interesting protein because it consists of multi-von Willebrand D domains, where most of them are cleaved autocatalytically, creating an anhydride at the new C-terminus that can bind to hydroxyl groups or amine groups on other proteins. And I could show that this one is covalently linked to the Mac2 molecule because they're also located in the mucin granules, both of these molecules. So this could actually mean that this molecule could act as an additional cross-linker of the mucus, stabilize its function or its structure. So the main function then of mucus is to be this barrier. This is the secreted inner material, and this material is impervious to bacteria which reside in the outer layer. There might be some microorganisms that can go through, and of course then the tissue will respond to those, but in general the main part of the majority of the microbes should be kept at a distance. And this protective mechanism is also stimulated by the bacteria. So if you don't have any bacteria in the germ-free condition, you don't have any firm layer at all, more or less. While then in the conventionalized animals, you do have a mucus layer, or this firm protective mucus layer is stimulated, and you keep the bacteria at a distance. Other bacterial components, I think, can possibly travel through this or be transported through the mucus layer, but not the entire bacteria. And if we look at this in a different way, we have set up a technique where we use live tissue, explants, either from human biopsies or from animal intestine, mouse intestine, and we put them in this perfusion chamber, and then we allow the mucus to be secreted, and then we can put beads on top of this. So we put 0.2 up to 2 micrometer beads on the mucus, and then we do cell stacks with a confocal microscope. We can see here is the epithelium, and in the human case, we never see, in the control patients, we never see any beads traveling through the mucus layer. While in mouse, most of the time we don't see beads traveling through, but at some points, as you can see here, some of the 0.2 and 0.5 micrometer beads can get through, but never 1 micrometer beads. So this is impermeable or defense mechanism of this mucus layer. Another thing that we have done that is quite interesting is that we have looked at the O-glycosylation pattern of Mac2 in human sigmoid colon. And the striking thing is that this graph represents is that we are very uniform. You see that there isn't any difference, even though we have different blood group, patients with different blood groups here. Normally in other epithelium and other tissues, the mucus glycosylation differs with blood group, for instance. But in colon, it seems like we are completely uniform. And the question is, why is that? Is this in some way a way to select for the flora, or is it maybe the flora that is shaping our glycosylation? I don't know. But it might be coupled to the microflora, I would think. So, now told you that the mucus is important in protecting us. So what if we don't have mucus? In the Mac2 deficient mice, we do not have mucus at all, and they develop spontaneous inflammation and cancer. And in this case, we have bacteria in contact with the epithelium constantly, so we can never clear these signals, and therefore, of course, you elicit inflammation. But if we don't want to use a model that is so dramatic as completely lacking mucus, we can turn into altering the mucus. And this has been done by Leon Xia in Oklahoma, and we collaborate with him on this, where he has knocked out different core structures of the O-glycosylation, which will affect the mucus. And in this core 3 knockout animals that lack an enzyme transferring this glucnac to this initial galnac that is linked to the protein, this is a structure that is not so common in mouse, and they do not develop spontaneous colitis, but they are more sensitive to DSS. This has been published. And then they have also developed the core 1 structure or knockouts, which lack instead an enzyme transferring galactose to this position. And these animals, this structure is much more common in mouse, and these animals do develop spontaneous colitis. And then we have also looked at the double knockouts lacking both these enzymes. And we then look at the bacteria in red here and mucus in green. We can see that the mucus layer is stratified and nice in the wild types, both at 3 weeks of age and 12 weeks of age, while the core 1 knockouts seem to have some kind of disorganized mucus layer at 3 weeks of age, but it is disrupted after 12 weeks. And in the double knockout, it's completely disrupted, and they are quite inflamed already after 3 weeks, so before weaning. And these effects seem to be depending on the bacteria, because if you treat them with antibiotics, you can see here that this normally, with that bacteria, histologically inflamed, very inflamed tissue is more or less normalized when you treat with antibiotics. And then I also looked at what could the mechanism be for this. And one idea is maybe that it has to do with Mac2 degradation. And if you think of this as the wild-type molecule with a lot of glycans on, you can think of this as the double knockout lacking a lot of the glycans, so it's not at all as protected. For the Mac2 staining, you can see with and without antibiotics that you do have a mucus layer. But for the double knockout, you can see that there is no mucus layer when you have the bacteria present. But there seems to be something adding on to the epithelium when you use antibiotics, so you remove the bacteria. And then I've looked at stainings for terminal fucose, so looking at one of the sugars that could be exposed. And in the normal case, we see that the mucus layer is stained with this, both with and without antibiotics, meaning that the compact structure of the mucus is not affected. These structures cannot cleave off in any extreme extent. While in this double knockout, we can see that if we don't have bacteria, we do have a secreted mucus that still contains these structures. But if you have the bacteria there, you don't see anything at all that is secreted, meaning that this sugar is already digested by the bacteria and probably then exposing more. And I would suspect, even though we haven't been able to show that yet, that also the protein core is digested. So now I'm turning into another story, short story, which is a model of colitis that I think that most people are quite familiar with, the IL-10 knockouts. And we looked into the mucus in these, and in our facility, our IL-10 knockouts do not look very inflamed. I wouldn't say that they are not inflamed, but they are not severely inflamed at least. And we have looked at quite young animals as well. So we started to look at the mucus secretion or the mucus growth, the thickness. And we couldn't see any difference in the mucus thickness in IL-10 knockouts and wild types. But then we started to look at the permeability. And in the normal case, you can see we have the epithelium here and we have the beads up here. So the mucus is separating nicely. But in the IL-10 knockouts that have as much mucus as this one, the beads are coming all the way down to the epithelium. So it's completely permeable. And also, if you look for the bacteria, just stained with DAP in this case, we can clearly see that bacteria is coming down into this not so well organized mucus layer that these animals do have. So it is not the thickness of the mucus that is of importance, but the quality of the mucus seems to be of importance here. So how is this then translated into human disease? And I'm mostly interested in ulcerative colitis. Since it is restricted to colon, it starts distally, where I think that the mucus protection is of most importance, and that it is a reaction to the normal flora, and a lot of this actually mimics the Mac2 defects that we see. So we looked a bit into patients. Firstly, we have looked into the patients, the glycosylation of the patients, and we can see that in active disease, we have a shift of several of the glycans, leading to shorter glycosylation. And maybe these shorter glycans are then more easily digested and more degradation of the mucus occurs. Well, this is then reversible since the non-inflamed tissue or patients do not show this pattern. But maybe this could also be affecting the chronic inflammation, since if it is more degraded, bacteria can continuously access the epithelium, and therefore the inflammation could be sustained. So if we look then at permeability, and this is ongoing actually, so I'm just going to present you a control here, which then nicely separates the epithelium, which I don't know if you see, but I can promise you that it's down there, and the beads up here. But then in some of the UC patients, definitely not all of the UC patients, but in some UC patients, we do see permeability of the mucus. So this might be one mechanism of this defective barrier or causing the disease. Okay, so I've talked about inflammation. What about inflammation that is caused by normal flora? But what about pathogens? And we don't have so many examples yet, but I will just show you this example, which is the Entamoeba histolytica, that has developed a way, because it's a colon pathogen, and in colon the pathogens need to circumvent the mucus barrier. And it has evolved developed a very special protease, a cysteine protease, that has a very strange cleavage specificity for Mac2, and it cleaves at this location, which is just next to the highly glycosylated regions. And therefore, this C-terminal part is cleaved off, and the dimer is then released, and the mucus is dissolved, which can be seen here, that we have the insoluble mucus, but if you treat it with this protease, it dissolves, and if you add an inhibitor, you still have the insoluble mucus. So if the pathogens then can dissolve this, and then the mucus is dysfunctional, and you will get inflammation or get infection. What about the commensals? Can they do this? We have just very recently looked into a little bit on this. And we looked at lactobacillus and bifidobacterium, and they do not seem to be able to cleave with this, which is then a fusion protein of the C-terminal part of Mac2. So it's not the whole molecule, it doesn't have the right glycosylation, so it's just a portion of the molecule, because it's very difficult to express these as full-length molecules or purify them and understand the data, I would say. But this fusion protein is then not cleaved by these bacteria. But if we take just an oral or fecal sample with mixed bacteria, we do get some kind of cleavage, indicating that there might be some microbes of the commensal flora that actually is able to cleave to a certain degree, at least, some of the Mac2. And maybe if you have a little bit too much of these, you will have a less good barrier that protects you. But this is something that, of course, needs further exploring. So, to summarize, formation of the protective mucus layer is needed to avoid bacteria, but also is also stimulated by the bacteria. Maybe there is some degradation from some bacteria, or bacteria that can turn on some proteases that will affect the mucus layer. And a defective mucus layer will allow bacteria to penetrate. So this can be in many different ways. The problems in the mucus layer can be in very many different ways, and I'm especially focusing just on mucins and the mucus, and not all other components that also, of course, could be a problem. But there could be mutations in Mac2 or other components making up the structure of the mucus. The biosynthesis is very tricky. There's a lot of cysteines in these molecules, and therefore any mistake in biosynthesis will lead to some ER stress and problems, and you will also get less mucus. So this is also a sensitive part. Defects in secretion, because if you have the wrong types of secretion, you will also not expand the molecules correctly, like in the CF phenotype, for instance. And the cross-linking, both with Mac2 itself and also with other components like the Fc gamma binding protein, could have effects on the structure of the mucus. Is it thinner? Is it less dense? Is it permeable? All these questions or these problems could lead to problems for or lead to bacteria coming in here, as well as then bacterial protease and the bacteria that could also harm this barrier. And then this bacterial will lead to bacterial contact, and maybe that could be one of the causes for ulcerative colitis. And I would like to acknowledge the Gothenburg group led by Gunnar Hansson, our collaborators, especially then Leon Sia and the gastroenterology unit where we get all our human samples. And of course we need support.